Okay, welcome back everyone. It's Dr. Lindner. Okay, in a previous video, we had mentioned and gave a little bit of a breakdown into the neuromuscular junction. Now, we're going to talk about the different types of muscle proteins. Now, we have contractile proteins, and you've heard me use these terms, actin and myosin. Actin and myosin are the contractile proteins in which calcium is going to have some sort of impact on to make the muscle contract. We're going to get into that in this video. What is calcium's role here? The regulatory protein, we have tropomyosin and troponin. And again, calcium is going to have some sort of impact on these two types of proteins. The structural proteins are called titin, nebulin, alpha-actin, myosin, and dystrophin. These are more involved in the alignment, the elasticity, and the stabilization of the muscles. Now, down below, on the right-hand side in B, we have the thin filament. This is actin. And off to the left is the thicker filament, number 2, which is myosin. Now, one of the things that I want to point out here is when we look at myosin, we have these myosin heads. And at the very, very tip, I'm going to circle it red here, we have a binding site. It's called an actin binding site. That's what this is here. And that's what that tip is right there. They're an actin binding site, which implies that this tip of the myosin head has to articulate somewhere on actin. I mean, it's called an actin binding site. Well, it's going to, or looking for, this location right here that I'm circling, called the myosin binding site. These are these little black circles that we see partially. We see it partially because there is a regulatory protein called tropomyosin that's covering it. And then we have this protein here called troponin, these blue staples, and that's what I want you to think of them as. Troponin are these staples that are holding the string, which is tropomyosin. It's holding, the troponin is holding the tropomyosin in its place, and its place is covering the binding site. Because if the binding site was not covered on actin, then the myosin would have a place to attach. Keep that in mind. So the sarcomere, which is the functional unit of muscle, is made up of these regulatory proteins called troponin and tropomyosin. They're basically the on and off switch. We have contractile proteins, which are actin and myosin. And again, the structural proteins that are involved with alignment and stabilization and elasticity of muscles. So these are the different types of proteins that make up the functional unit of muscle called the sarcomere. So when we look at actin, which is here, these purple little pearls, right, these spheres, notice that we have these binding sites. These are these circles or these targets. But we don't see the whole thing because of tropomyosin. That's the orange string, which is the regulatory protein that's covering the binding sites. And then we have we have troponin, which in this picture, these yellow ones, and the other picture they were blue, but here the troponin, these yellow staples, are holding the tropomyosin in place. Now, if something came along, like a staple remover, and pulled out these staples, If something came along, like a staple remover, and removed the staples, then tropomyosin has the freedom of rotating or shifting or moving. And when it shifts, these binding sites become exposed. Well, what is that staple remover? That staple remover is calcium. That's really important. The staple remover is calcium. So when the sarcoplasmic reticulum, that blue meshwork, releases calcium, what's deep to it are the actin and myosin. In these myofibrils, we have lots of actin and myosin. So what's happening there is now calcium plucks out the troponin, the tropomyosin shifts, and now these binding sites become available for these myosin heads to attach. So here are the myosin heads, and you see that there are many, many, many different myosin heads here. And they're going to have the ability to attach to those binding sites, right? Like a magnetic attraction to actin. So we'll put an A here for actin, and here is myosin, and these are the binding sites. Before, we had that string that was blocking that, and we had troponin that was holding it in place. But calcium came in, removed troponin, so tropomyosin shifted, and the binding sites were exposed. So again, on the top, we have troponin, we have tropomyosin, and we have the active site, which is the same thing as the binding site. And when calcium comes in and removes troponin and tropomyosin shifts, down below here at the bottom, we have these myosin heads that have the ability of flicking, because there's a hinge joint right there. So it can move. Once it attaches, it can flick, sliding actin. When it slides the actin, it creates a contraction. So on the top, we have a sarcomere at rest. You can see this H zone, this space here. We see how wide it is, but look at the H zone down below. It moved closer together because a contraction took place, because all these myosin heads attached to the actin, and they flicked it, moving the actin, sliding it inward towards the center, making the H zone much, much smaller. So the steps that initiate a contraction, here's the axon, here's the axon terminal, or the synaptic terminal. We have the end bulbs. We have acetylcholine that is being released through exocytosis, and remember, it only does that because calcium influxes inward, tells the acetylcholine to release through exocytosis in the synaptic cleft. It binds to the receptors. It goes down the T-tubule because it binds to that acetylcholine receptor. When it does, the sodium rushes in. And when sodium rushes in, it creates an action potential. It goes right down the T-tubule, transverse tubule. It articulates with the sarcoplasmic reticulum, right? It attaches right there. The sarcoplasmic reticulum releases calcium. The calcium is here. Now it's going to affect the sarcomere, where we have actin and myosin. Now it's not showing troponin and tropomyosin here, but based on what I told you before, when calcium rushes in, it removes the troponin. So tropomyosin shifts, and now the binding sites, or the active sites, are exposed, and it creates a cross bridge. A cross bridge is the connection between myosin and actin. They call it a cross bridge. And then it flicks, right? That hinge joint right there, that hinge flicks actin and moves it in that direction, creating a contraction. And the only way that it ends is by removing the acetylcholine. In order to end the contraction, the acetylcholine has to be removed. So to remove the acetylcholine, it uses an enzyme called acetylcholine esterase, the E for esterase. When there's no more acetylcholine, well, then there's no more excitation going down the T tubule. There's nothing stimulating the sarcoplasmic reticulum to release calcium, so calcium goes back into storage. And if there's no more calcium, then troponin and tropomyosin go back to their resting place, and the contraction ends, and you get this relaxation. So these steps here, 1 through 5, go along with these steps here, 1 through 5. And then 6 through 10 on this slide go over the steps that end a contraction. But one of the things that I wanted to point out to you, and you could replay this and read through these steps, but it is basically what I just mentioned. The only thing that I wanted to point out was right at the very end, eventually these myosin heads need to detach. And the detachment requires ATP, which is energy. So it says the contraction begins as repeated cycles of the cross bridge, binding and pivoting, and detachment occurs that is powered by the hydrolysis or the breakdown of ATP. And the reason why that's important is if you've ever watched like CSI Miami, Crime Scene Investigation, CSI New York, whenever they come across a homicide and they find a dead body, sometimes the body is in a state of rigor mortis, where the body's in a shortened, contracted state, where the fingers are bent, the wrists are bent, they're in fetal position. So their bodies are kept in that shortened, contracted position, because when they're dead, their body can't make ATP, it can't make energy. And you need energy to detach those protein fibers. Okay. Now, eventually, after several hours, the body does release these degrading enzymes that just break down all protein. But they can tell about how many hours or how long a body has been dead based on rigor mortis or not. Okay. And then the ACHE is acetylcholine esterase. This is the enzyme. Anything that ends in ACE is an enzyme. That's the enzyme that breaks down acetylcholine. Okay. Okay, we'll take a break. When we come back, we'll talk a little bit. You know what? I'm just going to, before I do take a break, just going along with the sliding filament theory of actin and myosin pulling towards one another. Myosin pulls on the actin, and it causes that thin filament to slide inward. Okay, and thanks to the structural proteins, there's a transmission of force throughout the entire muscle, and it results in the whole muscle contracting. So when we look here at the top, I want you to see, we see that calcium was released, and what ended up happening, if we look at actin, we see the binding sites are all exposed. You can see troponin and tropomyosin have been affected because of the presence of calcium, but these circles, the binding sites, are exposed, and that's why the myosin head is reaching up for actin. But one of the things that I wanted you to see here was that we have ADP and another phosphate, right? How this works, you have adenosine monophosphate, AMP, and if you add a phosphate to it, you get adenosine diphosphate, and if you add another phosphate here, we get adenosine triphosphate, okay? And whenever you see a phosphate, it's usually what we call CP, which is creatine phosphate, right? When the body has lots of ATP, the body doesn't want to keep lots of ATP, so we have the ability of taking ATP, removing one of these phosphates. If you subtract 1 from 3, you get 2. You get adenosine diphosphate. So we get this ADP plus CP, which is that terminal phosphate that we already removed. It's just that phosphate by itself, we typically refer to it as creatine phosphate. Okay, so keep that in mind. So now, over here to the right, look at the positioning of the myosin head here, and look how it changed here. Remember the hinge? It flicked. So it created this power stroke moving actin in this direction. Okay, now that it did that, we want this myosin head to be able to be released so it could be free to make another contraction. And the way that it does that is that it takes that ADP and that creatine phosphate, it combines them to create ATP, and it's going to use that ATP to detach the myosin head. That's the take-home message there. And then when it does, the ATP is hydrolyzed or broken down back into ADP and that additional terminal creatine phosphate. And when these join forces again, you can get ATP. So it's that continual cycle going back and forth.